What's up, Bio class? This is section 3 in our evolution unit. We're going to talk today about genes, variation in evolution, as well as genetic change. A lot of this information is conceptual and requires a little bit of understanding as well as memorization. Listen to what I say so that you can kind of get maybe a better definition of what I have written down. So by the end of this video, you should have achieved these learning objectives. Number one, identify what a population is. So we have a definition for that, and how it defines a gene pool. Number two, what is the relative frequency of alleles? So frequency is the amount compared to others. So you could say, what is the frequency of blue eyes in this classroom? That would be an amount compared to a total population. Next, what are the two sources of genetic variation? So how do we look different than everybody? How do we get that genetic variation? Why is a single gene more likely to evolve as compared to multiple genes? How does genetic drift affect evolution? So that's one of those concepts. And then last but not least is define the 5 factors that affect the Harvey-Weinberg principle. So hopefully by the end of this video, you can identify all these questions. Again, I put these learning objectives in to guide you and to kind of help you get the most out of this video. All right, so let's start out with variations in gene pools. So what I want to do is I want to define to you guys what is considered a population. A population is a group of individuals of the same species that interbreed. We are considered a population in Sheboygan Falls. We're considered a population in Wisconsin. But in the animal kingdom, if you're looking at turkeys out in a field, or deer that share the same woods, or fish that are in the same pond, that is a population. So if it's the same species, okay? Now, a gene pool results from interbreeding. So what that means is, you might have a pond and you drop 5 species of trout in there, okay, or 5 trout that are the same species. They're going to interbreed, and whatever genetics they have are the only ones that are getting passed around, unless something comes in from the outside. But we're not assuming that. So a gene pool results in interbreeding within a population. So this pool of genes is going to include all the different alleles that are present within the population. So, for instance, if we have a classroom filled with people who all are homozygous dominant for brown eyes, and we all interbreed, there is no way at all that any future generations are going to have anything but brown eyes. Why? Well, that's what's in our gene pool. Our population only has brown eyes. So understand what a gene pool is and understand what a population is. All right, next up, we talked a lot about probability in our previous unit. One of the things that we see, and I see that my slide is covered up here, so I'll kind of help you fill it in. Genetics really rely on probability, and we help understand sample populations by looking at the relative frequency. All right, so the relative frequency of an allele is the number of times that allele can occur in a gene pool. That's what you're missing here, compared with the number of times other alleles for that same gene occur. So what that means is, let's take a look. Here we have a sample population of mice. 48% are heterozygous black, 16% are homozygous black, 36% are homozygous brown. So this is your heterozygous group, where clearly black is the dominant allele. All right, so we could almost say that, take 48 plus 16, which would be 64. 64% of the population has black mice, whereas 36% are brown. But 48% are heterozygous. So why is that important? Well, we can predict, how often that brown allele that's not being expressed in some of those mice because it's recessive is present. We can look at the relative frequency, the amount of each. So basically, you look in the room right now and you go, all right, we have 30 kids in here. 15 have brown eyes, 10 have blue, and 5 have green. That is a relative frequency of an allele. Allele, again, is the different type of gene genotype for that phenotype, which means that if we're talking about eye color, which would be the gene for eye color, the alleles are brown, blue, and green. Evolution is considered any change in relative frequency of alleles in a population. So, if we start to see this brown number rise and this black number drop, we would classify that as evolution, which means that, these specific mice, for some reason, there's something increasing their fitness being brown. Maybe it's their environment, but the black ones are getting eaten, they're dying, they're not surviving, their fitness is low, the brown ones proliferate, and you might get to a point where there's 100% brown, which means that evolution occurred. All right, so what are two sources of genetic variation? This is a total review slide. Number one, your number one source is a mutation, which simply put is a change in DNA. And that can occur from mistakes, which we talked about in replication, or exposure to radiation or chemicals. So mutation is a number one example of a source of genetic variation. What's going to happen? Well, it can affect an organism's fitness. And you guys learned that some mutations are positive, some are negative, and some are just neutral. So by me saying that it can affect an organism's fitness is basically making the comment that it can be positive, negative, neutral. Many mutations do actually produce changes in phenotype. So what that is stating is that whether it's positive, negative, neutral, you might physically see a change, if we're talking about long periods of time, which is evolution, in that phenotype due to mutation. The next source is gene shuffling, which again, this is a review slide. It basically says how the DNA is recombined. And you guys know from prophase 1 and metaphase 1 in meiosis that we can have independent assortment and we can have crossing over. Those are, two examples of gene shuffling. So really what we could say is that there are three sources of genetic variation: a mutation, independent assortment, and crossing over. All right, so natural selection is something that I talked about in my previous video, which basically speaks to the fact that, certain genetic information is going to be passed on, and it's going to be selected due to the environment, not artificially. So it just means that, brown-eyed people in here, for some reason, you're going to survive better than blue-eyed people. So over the course of time, the brown eyes are going to be selected for, which means we're going to have less blue-eyed people, more brown-eyed people. That's all it means. Now, that's a single gene. And natural selection on a single gene can lead to major changes in those allele frequencies. So basically what I'm saying is that it's a lot easier to explain the theory of evolution based off a single gene than it is off a species. Okay, so below I have an example of the effect of color mutations on a lizard. All right, so the initial population saw 80% brown, 10% red, and 10% black. The next generation saw 80% brown and 0% red and 20% black. So what can you guys think would have happened? Well, let's put, a lizard out in a forest, and your predators are hawks, your predators are, ground-dwelling animals like, we can say snakes, or other rodents. You're going to see that red lizard. It's going to stick out like a sore thumb. So this red lizard potentially could have been a mutation, and it survived in one population, but it got killed off, and it's just gone. So that's a single gene trait that can lead to allele frequencies. It was 80-10-10. Now it's 80-20. That's an allele frequency change. We have gone from, 3 separate alleles to 2. All right? But notice what's happening. The next generation, so it could be 10 years later, 20 years later, 20 generations later, doesn't really matter. What this example is showing is that brown dropped and black increased. So over time, there might be some benefit where these black colored lizards are being preyed on less. And because they're being preyed on less, their fitness increases. Their survival increases. They can reproduce more than brown ones. Their genetics are passed on. So this is where we see this shift. And it all just happens because single allele traits are easier to see, and they can lead to overall changes in these allele frequencies, and thus we can classify it as an evolutionary change. Ooh, big slide here. Let me see if I can simplify it for you. Genetic drift is not a complicated thing. I have a lot on here, but it's really not that complicated. And I want you to think back to our laws of probability. We were rolling the dice, with the sperm and the egg example about a unit ago. And recall that genetics is controlled by probability. However, smaller populations are less likely to hold these laws true. Like I said before, there are 5 fish in a pond. The laws of probability are not going to be that great. This kind of random change in an allele frequency is called genetic drift, and it's strictly due to a small population. Okay, so these small populations, individuals that carry a particular allele, may leave more descendants than other individuals by just chance. What that is saying is that if you have 5 trout in a pond, all right, and 3 of them are females, 2 of them are males, one male might fertilize all of those 3 females' eggs, and the other one just won't. Genetics, like I said, that probability, that law was just broken. It was just proved wrong, because it would state that those 2 males would equally get an opportunity to fertilize those 3 females, but they can't, because genetic drift would state that randomly one of them just didn't. Okay, so over time, a series of these random chance occurrences will cause an allele to be common. Okay, here's a great example of genetic drift. Here on the ground, we have 6 brown beetles and 3 green ones. Somebody steps on 2 green ones. Now there's just 1 green one. All right, this just lowered the fitness of green bugs. It lowered the chance of passing those genetics. It lowered the chance of having a green bug ever again. That's why we have so many issues with getting endangered species. All right, when you start removing species from a population and out of that food web, you cause allele frequencies to change. And thus, you are actually creating evolution. So it's a tough concept, but genetic drift, if you can understand this, it's good. Here's another example of, where we just have that drift to change and it's just random. We're almost done here. Hardy-Weinberg, big principle in evolution and genetic equilibrium, and it states that an allele frequency will remain constant unless one or more factors cause that. So there are 5 factors I'm going to go through on the next slide, but it's basically saying that if these 5 factors don't happen, nothing will happen. The frequencies won't change. So going back to lizard, if it was 80-10-10, that's not going to change unless something happens. The situation in which allele frequencies remain constant and no change occurs is called genetic equilibrium. That's what equilibrium means. Now below, you don't have to write this down. We're not going to use it, but that's how it works. P2 is P squared would be the frequency of homozygous dominant genotypes. 2PQ would be the frequency of heterozygous, and the frequency of homozygous would be Q squared. So it's basically squaring the 2 homozygous genotypes and adding the frequency of the heterogeneous, and that should equal 1. Again, not to worry that you should understand that. So what are these 5 conditions to maintain genetic equilibrium? Number 1, you got to have random mating. So if you're going to try and maintain genetic equilibrium and not have any changes in your population, you have to have random mating, which means everyone has to mate randomly. You got to have a large population. Remember I said in a small population, probability gets screwed up big time. You can't have any movement into or out, meaning that if there are 5 fish in the pond, you can't add another one. Otherwise, you're going to have change, and one can't leave. You can have no mutations, and there can be no natural selection. So my question to you is, do you think all 5 of these things are always happening? Absolutely not. Sometimes, like look at gorilla populations. There is one male gorilla that will mate with all the females. Okay, that's not random mating. You might have that gorilla might be kicked out of that population. Well, now there's a new one. You're going to have mutations, and you're going to have natural selection. So all of these conditions have to stay stable in order to have no change. But what I'm trying to get you to understand is that over the course of millions of years, this stuff happens. Random mating doesn't happen. You might have a small population. Stuff will move in, or species will move in and out. Mutations will occur. Natural selection will occur, which is basically supporting this theory of evolution over time. So these factors are really, really important in truly understanding what's going on in this unit. So bounce back to those learning targets. Like I said, you should definitely have a good wrap on all 6 of those that I had up there, and hopefully you understand section 3. If not, pop by your local bio teacher or feel free to talk to me. Other than that, Yago's out.